Thank you all for standing by and welcome to our webinar entitled From Habs Place in the Water Column to Their Individual Cells. This webinar series called Freshwater Science will highlight Ohio Sea Grant and partnering scientists every month. Every quarter is a different focus, from human health and farm fishing to harmful algal blooms and human decision making, bringing applied research to the public on issues that affect our Lake Erie communities. I am Jill Gentis-Banicki from Ohio Sea Grant and Stone Laboratory, and joining me today is Dr. Tom Bridgman from the University of Toledo. Dr. Bridgman is the director of the U Toledo's Lake Erie Center and professor of ecology at the University of Toledo. He has conducted research on the ecology of the Great Lakes for nearly 30 years. Focusing on harmful algal blooms since 2002. Dr. Bridgman co-chairs the Ohio Harmful Algal Bloom Research Initiative, which consists of more than 30 science teams at universities across the state, seeking solutions to address HABs in Lake Erie. Dr. Bridgman created a new method to measure the size of the bloom each year and contributed to the development of the annual HABs forecast and metrics for determining the official impairment of the lake. His laboratory tracks bloom development and provides early warning and advice on bloom movements for water treatment plant operators throughout the HABs season. We're really delighted to have Dr. Bridgman here to talk about his HABs research. Before we do, just a few things about the logistics of the webinar. During our presentation, all participants are in a listen-only mode. Afterwards, around 12:20, I'll conduct a question and answer session. If you'd like to ask a question during his presentation, please feel free to pull up that chat feature anytime during his talk, and I will collect and pose your questions out to Dr. Bridgman at the end of his presentation. As a reminder, this webinar will have auto captioning and is being recorded to be posted onto our website for later viewing. Also, we will post a webinar survey in the chat feature toward the end of the half an hour. Please take a few minutes after the webinar to fill out that survey. It always helps us continue to bring you great webinars. So without any further delay, I'd like to introduce Dr. Tom Bridgman, presenting from HABs Place in the Water Column to Their Individual Cells. Dr. Bridgman.
Thank you, Jill. Can you hear me okay?
Absolutely.
All right, I'm going to start to share my screen and see if I can go right into. So does that look good for you? Or are you seeing the present?
No, I'm not seeing it in presentation mode yet.
How about that? Is that better?
There you go. Yep.
Okay.
Perfect.
All right. Well, thanks everybody for joining us on this lovely Thursday lunchtime. Hopefully you're at your desk or at home having some lunch and getting a little science in along the way. Just want to acknowledge a lot of people who have participated in this research. My collaborators in Germany, Christian Moldanke, in the Department of Engineering here at the University of Toledo, Kopei Tsai, Zach Kirschman, and Youngwoo So, and then my former graduate student, Eva Littmer, who did the vertical movement study. So my talk today is going to be in two parts. I'm going to spend most of the time talking about cells and how they break open and release toxin. And then I'll talk a little bit about the vertical movement of harmful algal blooms. And both of these are relevant for water treatment. We are located, if you're not familiar with the Lake Erie Center, we're located right on the shoreline of Lake Erie, right next to Maumee Bay State Park in Oregon, Ohio, right on Maumee Bay. And we are highly impacted by HABs. And our drinking water comes from right offshore in Lake Erie. So we are often asked questions about drinking water and how can we help the drinking water utilities in Western Lake Erie. So that's what this presentation is focused on, largely on helping drinking water plants. So I want to start off with part one, which I'm calling When HABs Go Bad. And just to sort of go over some fine points here of introductory before we get started. when I talk about harmful algal blooms or cyanobacteria, in this case, I'm specifically talking about Microcystis. That's the species that we get in Western Lake Erie. That's the one you see on the satellite photos and in most pictures. It's that bright green stuff. So we did work with Microcystis. The toxin that is inside Microcystis is called microcystin. So if I say toxin, I really mean microcystin specifically in all of its hundreds of congeners or forms that it has. The other vocabulary term is intracellular versus extracellular. When I say intracellular, I'm talking about toxin that is inside the microcystis cells. So that's inside is intracellular. If this toxin gets outside of the cells and is released free, dissolved into the water, we call that extracellular. So I'll use those terms a lot. So this study really focuses on those two forms, intracellular versus extracellular toxin or microcystin. So why do we care? Why do we care where the toxin is? After all, it doesn't matter if your dog is unfortunate enough to go down to the beach and lap up some toxic water at the beach. It doesn't matter whether the toxin is inside or outside the cells. The dog is getting the same dose of toxin. And most of the time, the toxin is inside the cells. About 90% of the time or more, or 90% of the toxin is inside of the cells. So there really, most of the time, isn't much extracellular toxin. So why do we care about it? Why are they important? It's because when those toxins do become extracellular, it can have an impact. It can be an impact on inhalation of toxins, because as we're finding out, the aerosolization of toxin through wave action and wind, aerosolization of toxin is becoming more a prevalent source, or we're knowing more about it as a source of exposure. And it's going to be those extracellular toxins in the water that are aerosolized. And so that's important for people who might be at the beach or windsurfing or something during a bloom. And it's also important for drinking water treatment, whether the toxins are extracellular or intracellular. And that's mostly what I'm going to be talking about, is drinking water treatment. So as the figure shows, there are intracellular toxins. The microcystin is inside the cells. Extracellular, it's outside the cells. And most of the time, 90% or more is inside the cells. So let's go on. Let's talk a little bit about drinking water treatment. And hopefully there's no drinking water treatment professionals in the audience, because I'm not a professional, and so I might make some mistakes. But generally, this is, at least for the Toledo area, this is the drinking water process, is to, as the water is coming into the plant, it is treated with permanganate and activated carbon. And the permanganate helps to prevent zebra mussels from clogging up the pipes. And both the permanganate and activated carbon can help remove any toxins that might be dissolved into the water. But the main way that toxins are removed is by removing the cells intact. So, the process is coagulation, flocculation, and filtration. I'll just refer to it basically as filtration. The microcystis cells are filtered out intact so that they take the toxins with them. There's always going to be a little bit of extracellular toxin, and that's taken care of through treatment, chemical treatment by chlorine, ozone, bacterial breakdown of that extracellular toxin. But I think I have so an easy way for me to picture this is I picture a swimming pool full of water balloons, and the swimming pool water is our drinking water, and those water balloons are the microcystis cells full of toxin. So if you were the person in charge of this situation, you would want to very carefully remove those water balloons without breaking them and spilling any toxin into the water. And that's kind of the situation that water treatment managers face. It's kind of a balancing act. They want to carefully remove those cells or those water balloons without breaking them open. Yet they do have to add some chemicals to the surrounding water, because there'll be a little bit of toxin in there. So they want to add enough chemical to to take care of any residual toxin in the water, but not so much chemical that the chemical breaks down the water balloons and breaks them open. So it's always a balancing act for water treatment managers. So just kind of keep this this example, this image in your mind of these toxin-filled water balloons. All right, so this takes us back to the water crisis of 2014, and I'm sure many of you have seen slides like this before, but I need to just refresh us a little bit. In 2014, we had the Toledo water crisis or incident in which microcystins in the treated Toledo water exceeded the World Health Organization guidelines, and there was a do not drink advisory issued, and approximately 500,000 people were without drinking water for a couple of days. So going back to 2014, what were the factors that are considered to have caused that that incident? First of all, we know of four factors. The bloom was located near the intake. So it concentrated around the Toledo water intake. It was a heavy bloom that year. So a large biomass of cyanobacteria. It was very toxic that year. It was for whatever reason that we don't understand, the bloom was highly toxic that year. And then there was vertical mixing. There was a lot of wave action that may have forced the bloom down into the water intake. But all of those factors alone should not have resulted in the water crisis, because the water treatment plant managers at that time were aware of the bloom and they were treating it aggressively, and they were doing all the things that they had been doing for the past 10 years before that, at least. So the question has always remained, like, why did the treatment processes that they were using in 2014 not work that year? What was different about 2014 that what they had been doing that had always worked before didn't work in 2014? So was there a fifth factor? And it turned out that it was kind of determined a couple of years later using water samples from 2014 that there was a fifth factor, and that was that there was a big outbreak of cyanophages in the lake that year. And cyanophages are viruses that attacked bacterial cells. The cyanophages attacked the cyanobacteria and broke open the cells and released the toxin into the water. And this was published a few years ago by a group from NOAA, Bowling Green, and other places. And so a broad-scale cyanophage infection may have lysed the microcystis cells. Lysed means breaking them open and causing a significant increase in the extracellular or dissolved toxin entering the treatment plant. Okay, so that may have led to the water crisis in 2014 because, as I said before, this is the usual way of treatment. You are removing the cells intact, and then you're just adding a little bit of chlorine and other chemicals to mop up whatever dissolved toxin is left over. But with the situation that they may have probably had in 2014 was this: the cells were attacked by viruses and the cells were breaking open. And now a lot of that toxin had left the cells and was dissolved in the water. So it was no longer being filtered out. Those dissolved toxins are not filtered out. They can go right through the treatment plant unless they are treated aggressively by chemicals. So if you look on either side, the amount of toxin hasn't changed. It's the same amount of toxin. It's just in a different form now. And if the managers are not aware that that has taken place, they may not know that even more chemical is needed to take care of all those extracellular toxins before they can get all the way through the plant. So this is probably the situation that we had in 2014, because it's very difficult to know whether the toxins are inside or outside the cells. That's a difficult thing to know in a timely manner to be able to change treatment processes. We'll never know for sure, probably exactly if that happened in 2014, but fortunately or unfortunately, a similar incident happened again in 2019. We had a large bloom, and I and others noticed in 2014, over a course of a few days, we started to see miles of foam on the water surface of Lake Erie. And this stretched all the way from Cedar Point all the way to Toledo, covering hundreds of square miles of the lake. There was this foam on the surface during the bloom. And that indicates to us that there's some cells are lysing, and that foam is being caused by protein that's being released from the lysed cell. So there was some sort of lysis event going on. And we saw that the microcystis became extracellular within 1 day. So that the figure on the right there is from the Toledo water treatment plant. And you can see they're tracking, this is day, every dot on there is 1 day. They're tracking the total toxin and the dissolved toxin. And it looks like on about August 4th, the toxin went from almost all inside the cells to about 80% outside the cells. So in just a very short period of time, the toxin went extracellular. So that fortunately did not result in a water crisis, because by then Toledo had an increased treatment capacity, and they were treating aggressively. And it was not a very toxic, fortunately, it wasn't a very toxic bloom that year. So that level of toxin release didn't cause a crisis. But if we had, we could have easily had 10 times as much algae out there, that year. And if that had taken place, we might have had a problem. So this is just to say that it's very difficult to know whether the cells are lysing or not. Daily or weekly measurements may not be enough. What you really need is some sort of real-time capability of knowing whether the cells are lysing and the toxin is going extracellular. So, so fortunately, that capability has been developed through some instruments developed in Germany. And we call this the early detection of cyanobacteria cells. And we use something, a component called unbound phycocyanin. And just to be slightly technical here, a lot of how we measure cyanobacteria and algae is through a technique called fluorescence. Fluorescence is where you shine a specific kind of light on the algae, and the algae starts to glow and shines a different kind of light back at you. And that's called fluorescence. And the amount of light that shines back at you gives you an indication of how much algae is there. So, and different kinds of light coming from the algae can tell you things about whether it's diatoms, good algae, bad algae. But there's this extra capability now called unbound phycocyanin. So if you look at the figure there, usually there's two main pigments in blue-green algae. There's the green pigment, which is chlorophyll, and the blue pigment, which is called phycocyanin. And they are bound together. But when the cell starts to become unhealthy or gets in trouble, the chlorophyll and the phycocyanin become detached from each other. And then you get this unbound phycocyanin, which has a different fluorescence signal. And instruments can pick that up instantly. And that unbound phycocyanin signal corresponds very closely to when the cell membrane is starting to break down and release toxin into the water. So can't measure toxin release directly, but you can measure this fluorescence signal that corresponds very closely to it. And this has been published in some studies using chemicals to break down the cells and then measure the fluorescence changes. But we wanted to see if we could do this with the viruses, if we could do this with the cyanophages. And so that's what we really focused on in our study here. Our approach was to deploy we had two instruments, one of them called the PhycoSense. We deployed it at the Toledo drinking water plant last summer, 2022, to monitor during the bloom season. And it was set to monitor every 15 minutes. And the signal went online and was available live on the web. We also did some laboratory experiments. So we know that that Mother Nature might not cooperate and give us a viral attack on the bloom every year. So we have to do it in the laboratory to make sure we can get some good results. So that's what we did. So here's the results from the Toledo water intake. This is last year. And the basic results are at the bottom there. You can see the different kinds of algae, cyanobacteria, green algae, diatoms, cryptophytes. That's from July 15th through about October 17th. And you can see that where the main bloom was there at the late July and into August. So we did see some unbound phycocyanin detections early in the season and then late in the season. There was a little pulse of unbound phycocyanin indicating that there was some cell lysis happening. So that corresponds with the top panel. There is the toxin measurements, and the ratio of extracellular to total toxin was higher at those same times. So indicating that that when we did see that unbound phycocyanin signal in the fluorometer, that yes, there was some toxin being released from the cells. In other periods, though, it was essentially a healthy bloom throughout the season. We didn't have a big viral attack. We didn't have a big release of toxins. So just early in the bloom, which might have been due to some zooplankton grazing, and then at the end of the bloom, it's normal at the end of the bloom every year for the cells to break down and release a little toxin, and that's what we saw in late September. Okay, so now moving on to the laboratory experiments. These were done using microcystis cultures, and this one is with hydrogen peroxide, which is a common algicide. And so we're also doing some studies on behalf of some inland reservoirs where they use algicide to treat blooms in their reservoirs. So this is relevant to them. The panel on the right, or I'm sorry, the panel on the left is the control. So over time, it's just an algal culture or a cyanobacteria culture that's growing well. You see it's growing, getting bigger, and all the toxin is inside the cells. There's no extracellular toxin. In the middle panel, we treated with some hydrogen peroxide, and you can see now you start to see gray bars. Those gray bars are the extracellular toxin. I wonder if I can move my mouse and it works. You see my mouse? So you see the extracellular toxin here. And sure enough, you see that red dot, that's the unbound phycocyanin, just showing that the release of toxin corresponds very well with this unbound phycocyanin signal. Again, this is hard to measure, or takes time to measure the toxin itself, but this unbound phycocyanin signal is instantaneous. It just, you can read it right away. If you add even more hydrogen peroxide, you get an even bigger unbound phycocyanin signal with that release of toxin. All right, so next figure, this is now using a different instrument called the PhycoSense. The PhycoSense is a flow-through instrument, and you can measure every 10 minutes if you want to. Again, on the left panel, this is a healthy microcystis culture. This is the control, so no additives or anything. It's growing nicely. Almost all the toxin is increasing, and toxin is inside the cells, and we don't see any unbound phycocyanin in that healthy control. Now, if we add potassium permanganate is added at the intake to control zebra mussels. If we add potassium permanganate, after 24 hours, we see a spike in unbound phycocyanin, and we start to see the toxin being released from the cells. If we add even more potassium permanganate, we don't see anything, because there's so much potassium permanganate that it destroys all the toxin, and it also destroys all the unbound phycocyanin. So this has some relevance because I mentioned before, there's this balancing act with treatment. If you don't add any potassium permanganate or chemicals, you might be able to remove all the cells intact. If you add a certain amount of potassium permanganate or other chemicals, you might break open the cells, but not have enough chemical to destroy the toxin. So the lesson is that you either want to add minimal chemical and keep the cells intact, or add so much chemical that it just destroys everything. You don't want to be in that in-between area of in the middle, where you're breaking up the cells but not destroying the toxin. And this this unbound phycocyanin can kind of tell you that you're in that uncomfortable middle area where you've added you're in the middle where you haven't added enough chemical. You've added too much, or but not enough to take care of everything. Okay. So this next panel, then we finally get to the viruses. This is where we added viruses to the culture to see what happened. Again, on the left is the control, nice healthy culture. On the right, though, you start to see about 48 hours after infection, you start to see the unbound phycocyanin signal. And then a day or two later, at about 72 hours, you see the increase in unbound or in extracellular toxin. So the experiment worked. We were able to lyse these cells, and we were able to get an unbound phycocyanin signal that corresponded to the extracellular toxin. So this is good news for the ability to be able to detect this in Lake Erie. Okay, so in summary of this part, we don't get those cyanobacterial lysis or cyanophage events every year. We've just really seen them in 2014 and then again in 2019. So maybe we don't have no idea how often they happen, but it's not every year. But when they do happen, it can have significant consequences for water treatment. The unbound phycocyanin signal provided by these new instruments is a good rapid indicator of when those cells are breaking open. And it can be informative for water treatment. The application of water treatment chemicals can help managers know whether they're adding not enough or too much water treatment chemicals. We did notice, however, that unbound phycocyanin signal is really ephemeral. It like shows up and then it disappears. So it's something you have to be watching for. You know, if you just went out and tried to measure it every day or two or once a week, you might miss it. So it's something where you really need to have a frequent measurement of, as an online sort of maybe every half an hour or so measurement. We've got some future studies going on this. We are studying additional cyanobacterial species such as Planktothrix and others that are more prevalent in inland waters and inland reservoirs. And then how well do these instruments and this signal work in challenging environments that are very turbid and cloudy, such as rivers and reservoirs and so on. So that's where we're going with this. I guess I have a few more minutes. I just want to acknowledge our funding on this comes from the U.S. Army Corps of Engineers and with help from the Great Lakes Center for Fresh Waters and Human Health at Bowling Green. And then I've got just a couple minutes. I want to talk about part two, which is the vertical movement of harmful algal blooms. And this was a study we did several years ago with my graduate student, Eva Kramer, and others. And this was funded by the Ohio Department of Higher Education's HABRI, Harmful Algal Bloom Research Initiative. And the question was, again, this is to help water treatment managers, especially Toledo and others who have intakes out in Lake Erie. When the cyanobacteria, this microcystis, is floating at the surface in a surface scum, does that mean there's less microcystis near the bottom where the intakes are? And if so, can the water utilities reduce their exposure to microcystis by timing the pumping according to vertical location of microcystis and related to wave action or time of day? Because we know the microcystis goes up and down in the water column. Sometimes it's the surface, sometimes it's spread out. Are there some patterns that we can use so that when it's all at the surface, or we think, is it all at the surface? Does that mean we can pump more where down at the bottom where the microcystis is not? So that was the question. And so we did some 24-hour studies. We tried to go out there in rough water, calm water. We did a few, like I said, 24, 48-hour studies where we parked the boat out near the water intake and measured every 2 hours. And here's just a couple of the results. So during rough conditions, we could see that there were the blue-green algae and diatoms were evenly distributed through the water column, but the green algae and cryptophytes were concentrated near the surface. At night, everything was evenly distributed. So it's, things are being mixed up from top to bottom. So if hopefully this figure is self-explanatory. It's sort of a cross view of the lake, cross-section view of the lake, and the different colors are the different kinds of algae. It might be not clear, but the blue dots are blue-green algae, the green dots are green algae, the magenta dots are diatoms, and the red dots are cryptophytes. So there's different kinds of algae, and they kind of they can partition themselves a little bit differently from top to bottom. But usually during rough conditions, they're sort of all mixed up. During calm conditions, we did see that the blue-green algae were not right at the surface, but they were concentrated a little bit down further from the surface and increased below a depth below 2 meters of depth. At night, when it was calm, the diatoms sank to the bottom. So diatoms are almost like little grains of sand. They sank to the bottom in calm weather during the day, whereas the other algal groups were all evenly distributed from top to bottom. So we didn't see a lot of change from other than diatoms, we didn't see a lot of change from day to night. So this is not telling us too much, but the bottom line, I think, comes in this next figure where, like, if there's a surface scum, does that mean there's less algae down at the bottom? That's the most basic question. And we found out, unfortunately, no. And here's a time series measuring it starting at 5 a.m. in the morning, 7:30 in the morning, 10 a.m., noon, 3 p.m., and so on. Most of the time, the blue-green algae, this is just the microcystis or the blue-green algae, it's evenly distributed from top to bottom. And right around noon, as usual on this calm day, we saw surface scum form. That's what the red circle is. You can see this surface scum forming right at the surface. This is top to bottom. But that doesn't mean that the algae at the bottom is going away. So the surface scum is a little bit deceptive. You think because you're seeing a surface scum that all that cyanobacteria is at the surface, but in fact, it's only a small percentage of the total microcystis that's forming that surface scum. There is still plenty of microcystis all the way down in the water column. So just because you see a surface scum doesn't mean that it's all at the surface. There's still a lot all the way down. And that was the bottom line that we came up with. So unfortunately, it doesn't look like water treatment managers can use this sort of vertical movement timing to time their pumping. And that's what we ended up with on this. So I think that is my last slide. And I guess I will stop there, and I will stop sharing and hand it back to Jill.
All right. Well, we have gotten some great questions during Dr. Bridgman's presentation. So let me get started and ask Dr. Bridgman as many as we can get to. I think we should be able to. So, well, okay. So the first question that we had gotten, and I thought this was a great one, was, can you explain why microcystis is such a significant member of the algal community? Is it nutrient balance?
That's a good question. And you know, there's a lot of factors involved in why microcystis becomes dominant. Microcystis and other blue-green algae are highly competitive. I mean, it's all competition, right? There's all these different kinds of algae out there competing with each other. And the question is, why does it become dominant? Blue-green algae, microcystis, and others tend to become dominant when there's a lot of phosphorus in the water. When there's plenty of phosphorus in the water, and the water is warm, they tend to outcompete other forms of algae. So high phosphorus levels, warm temperatures, that's when they do really well. Microcystis in particular, because it has this ability to regulate its buoyancy and go up and down in the water column, that gives it an additional competitive advantage. That is an advantage when the water is over a certain depth, like maybe, I don't know exactly, but maybe let's say over like 5 meters of depth or something like that. You know, when the water is really shallow, that buoyancy ability isn't much of an advantage, which is one of the reasons you don't see microcystis as much really in shallow areas like Sandusky Bay, Grand Lake St. Mary's, you know, in some of the rivers, is because that buoyancy doesn't give it an advantage in those situations.
All right. Another question we had was, did your findings show that there was a relationship between, we had quite a few people asking about water temperature and the presence of algae, what sunlight and the presence of a certain type of algae. Can you talk a little bit about those kind of factors?
Sure. I mean, we didn't study any of those factors specifically in the study, but in general, it's known that the cyanobacteria like warm temperatures, and the earlier that it warms up in the spring, the earlier that we'll start to see the cyanobacteria start to really ramp up. Sunlight is an important factor. We think that, bright sun, bright sunlight is an advantage for some cyanobacteria, but too much sunlight is also damaging. So again, the ability of Microcystis to regulate its position in the water column, if it's getting too much sunlight, it can kind of sink a little bit to get away from the sun. So, the cyanobacteria can find the right place in the water for them to get the right amount of sunlight, but not too much sunlight. So yes, their sunlight temperature is a very important part of it. Wave action and mixing, they all play into it.
Okay. Can you talk a little bit as to why Microcystis, why it wasn't a dominant algae in the 60s, but now it is?
Yeah, I think that is not really very well known. I don't know that anybody's really kind of solved that mystery. It could be that ratios of nitrogen to phosphorus have changed. There's some of the cyanobacteria that were dominant in the 1960s were what we call nitrogen fixing cyanobacteria. They can be competitive when there's not much nitrogen in the water because they are able to pull nitrogen out of the atmosphere. And so Microcystis cannot do that. So the fact that Microcystis has become dominant suggests that there's more nitrogen available now that allows Microcystis to outcompete some of the other cyanobacteria that were more dominant in the 1960s. So I think that's probably a nitrogen factor.
Okay. Let me see. We had quite a few, so I wanted to get some questions in about the first part of the research you talked about. Let me see. One question people were asking was, how strong is the correlation between unbounded, and I'm going to butcher the name,
Phycocyanin.
Thank you, and viruses, and how much confidence is there that the former can predict the latter?
That's a good question. I think what the unbound phycocyanin is really telling you is that the cells are breaking open. It doesn't tell you why the cells are breaking open. So you could be breaking open cells with chemicals or with viruses. So it doesn't tell you the reason that the cells are breaking open. And I think a lot is not, getting back to the viruses, I think a lot is not known about the viruses. I think the thought is the viruses are always out there, but there are some environmental factors that cause them to become active and then start actively replicating and destroying the cells. But I think that's really poorly understood. For example, we had the big viral outbreak in 2014, and so they were looking for it again in 2019, and they didn't see it. And the reason they didn't see it in 2019 is because it wasn't the same virus. It was a different virus in 2019 than it was in 2014. And the next viral outbreak may be yet another virus that we didn't know to look for. So kind of tracking the viruses is maybe not the best way to go. I think just directly measuring whether the cells are breaking open or not is a better way to go.
Okay. All right. So I have a couple questions dealing with the vertical movement research. The first one is, wouldn't the vertical movement be heavily dependent on winds like we had in 2014, given how buoyant scums are?
Yeah, winds will break up a scum, and if there's a scum formed and then a storm comes along, it'll break up the scum and it'll mix, depending on how strong the wind is, it will mix the water from surface to bottom. So wind action can always overcome the buoyancy of the microcystis and mix it all the way down. Yeah, in 2014, there were heavy winds from the northeast that were mixing the water from top to bottom. So I'm not sure if that answers the question.
Yeah, I think so. Another question that we had was, if is the cyanophage that attacks microcystis exclusive to microcystis, or does the same phage attack other genera also?
That's a good question. And yes, it is specific to microcystis. In fact, it's specific to specific strains of microcystis. We have the strain of microcystis that we were using in our laboratory came from Japan, and the virus was also came from Japan. We tried to use that virus on our Lake Erie microcystis, and it didn't have any effect. So there's a close match or close pairing between viruses and hosts, and so they're not broad spectrum viruses. They're very, very specific, even within a species.
All right. A question about the last slide that you had presented. On your last slide are the cells that are not in the surface scum during midday physiologically different. Sorry, I think there's a missing word here. Hold on a second. On the last slide are the cells that are not in the surface scum during midday physiologically different and perhaps more prone to lysis and microcystin release?
That's a good question. I don't really know that I have the answer to that. That would be, maybe that's an area for future study where we could go and take samples from various depths and examine them to see. One way to examine that would be to take samples from various depths and then to maybe add some treatment chemicals and see if some depths are more easily lysed by treatment chemicals than others. Yeah, that's a good question. At any given time, microcystis or any algae, it's going to be a population, and there's going to be outliers that happen to have gotten a lot of sun the day before and did a lot of photosynthesis, or some that were starved for photosynthesis. So it's just like any population. There's going to be variation, and so some will be able to rise to the surface, some will not on any given day.
All right. Do you have any idea how many waterborne viruses are in Lake Erie and how many can break open algal cells?
I have no idea. And I don't know that anybody does. That would be a good question for somebody like George Bullerjohn at Bowling Green. I think there are certain numbers of viruses that are being tracked. I know there's a lot of genomic work, genetic genomic work being done on Lake Erie. And so I think probably the answers to that will be forthcoming, but I'm not the person to
Okay.
be able to answer those.
Okay. Another question, and I can't remember, you said you have to be off by 10 till, is that right?
I have another meeting at 1, but
all right.
So it's like 2 till.
No, you can't do that. You can't rush like that. Okay. I have another question. Your mention of the relationship of microcystis to depth is interesting. Microcystis is certainly the most common that I've encountered in small ponds that rarely exceed 4 meters depth. Any speculation to why?
I don't know why. I don't normally see microcystis in ponds. It's usually something else. So I would be curious about some of these ponds that have a lot of, if microcystis is dominant in some ponds, I'd be very curious about that, because in ponds, it's usually something else. Microcystis does better in open waters with a certain amount of wave action and a certain amount of depth. So yeah, I'm curious about that. And, maybe the person can contact me and we can talk about it.
All right. We had a couple people asking about airborne toxin microcystis, and one person was asking, does exposure to aerosol microcystis always have toxic effects when humans are exposed?
I can't address the effects. I think this is a very new field. And there's a lot to be learned about the effects of airborne microcystin toxin. I know that airborne microcystin has been measured. There's air measuring devices that can measure it. I think it has been detected in people or in animals. But as far as what the health effects are, whether exposures have been large enough to have health effects, I don't know. I just know it's been detected.
Okay. Sorry, I'm kind of trying to go through these here. One person had asked if, and this might be out of your scope, but I thought it was an interesting question. Has any research been done for a machine or grinder that can lyse cells and be used in the field? The freeze dry thaw method for releasing cellular toxins isn't applicable in field settings.
Yeah, actually, I do know a little bit about that. If you were measuring, I don't know if they're talking about measuring toxin or trying to destroy cells or what they're doing. In order to measure toxin, you have to break open, you have to lyse the cells. And that's that freeze thaw method. You take a sample, you freeze and thaw it so that all the toxin can be measured, because the toxin test can't measure the toxin if it's inside the cell. So you have to do this freeze thaw method. There are other techniques where you add like these microscopic beads and vibrate them to kind of, it's called bead beading, where it's a technique in the laboratory to open the cells. Those don't seem to work very well. So the freeze thaw method is still kind of the best way that we have of lysing the cells. And you can do it pretty quickly. If you have some liquid nitrogen or something on the boat, you can dip the sample in there and freeze it, thaw it, and you can do it fairly quickly. But not everybody has liquid nitrogen or available. I don't know if that answered the question. Maybe the person can contact me if they had something more specific in mind.
Okay. Let me see here. I know that there's a lot of research trying to delineate the range of airborne microcystin. Any idea what the range is yet? Approximately 100 yards, miles?
I think there has been, there's been some research on that at University of Toledo and other places, with air samplers. But I don't know the range. I don't know the results of those yet. I don't know that they've been published. So I think those results are still coming out or still in the works.
Okay. Sorry, I'm kind of scrolling as I'm going through here. I think we've covered the majority of them. Sorry, I'm like just looking to see if I have any new ones that have popped up. I think that might be all of them. Wait, hold on one second. And it looks like Ji Young said 13 kilometers for the airborne.
Oh, that Young Lee. Hello.
Yes.
I'm glad she's there and could answer that question. Thank you.
It's always nice to get an answer like that,
Yeah,
isn't it?
Thanks, Young. Appreciate that.
Thank you. So here's my one of the last questions. So what is the next step to your research that you have going right now?
The next step right now is to move from what we found on Lake Erie into some inland reservoirs. We're working with Defiance, City of Defiance, their water treatment, their water reservoir that uses Maumee River water. Bowling Green's drinking water plant is on the schedule, and we want to see how well these instruments that we're using work in those situations, because the river water and reservoir water is very different from Lake Erie water. So we just want to expand on that.
And what are you seeing so far this summer?
On Lake Erie so
Yes.
far, I was out on the water yesterday. I didn't go far, but right now it looks good. There's still a lot of diatoms in the water, which is that good algae. So, I haven't seen any sort of big bloom activity yet, but we're scheduled to go out again early next week, and we'll see what happens. But so far, it looks like a normal year.
All right. All right. Well, I think this is a good spot to end.
Yeah.
So I want to thank you again, Dr. Bridgman. These are always great conversations. I really appreciate the fact that you came on and talked to us about your research. This was really a great discussion. I did also want to remind everyone that there is a survey URL in the webinar chat feature. So if you could take a few minutes to fill that out, that would be great. This webinar series is sponsored by Ohio Sea Grant and will continue on in July with Bowling Green State University's Kevin McClooney about his research on pharmaceuticals near treatment plants in Lake Erie's Western Basin. The registration link is in the chat. Thank you again, Dr. Bridgman. It's always great to have you talking about your research. We always enjoy it. And thank you to all the participants on this webinar. We hope this was beneficial and hope you'll join us again on an upcoming webinar. Thank you again, Dr. Bridgman, and everyone have a great afternoon. Thanks again.
Thanks, Jill. Thanks, everybody, for joining.
It was really great. Thank you.